Hey guys, Nathan here with the first of many videos regarding the second midterm in Biology 210. Last midterm I made a PowerPoint, an incomplete PowerPoint, which people said it was helpful to them, even though I uploaded it, a few hours before the test started, so, I don't know how much help it was, but I'm glad it helped. This time I've learned from my mistakes. I'm not going to be as lazy, and I'm going to start working on these videos and PowerPoints a little bit earlier. So this is each part kind of will consist of maybe 1 or 2 chapters, depending on how big the chapters are, and they should be approximately 20 to 30 minutes in length. Okay. Before I really get into the material, I want to say this. I'm only going to repeat it once for this video, so hear me out. You guys should study, especially for genetic stuff, especially for this organismal stuff. There's a lot of memorization, and I can talk to you guys all I want, and you can listen to Khan Academy and other places all you want. But if you don't memorize information and if you don't make a serious effort, you're going to have a bad time. On the test, we know that the test is going to be, very tight on time. Dr. Cap's pushing you guys. If you don't know this stuff, like the back of your hand, I don't really know the back of my hand very well, but, if you don't know this stuff like the back of your hand, I guess, then you're going to be in some trouble. So please do take some time and just memorize some stuff. It's not that hard. There's only a couple things that you really need to memorize. I'll cover most of the things that you need to memorize. So just, please be aware. Secondly, I'm definitely going to make mistakes. I make some really dumb jokes. Please bear with me. I apologize if I make mistakes. But that's how it goes. If you do catch me making a mistake, please comment on the video below, just to let other people know that I made a mistake. Things like that. Without further ado, let's get into this video. So, we'll be talking about Chapter 12, the cell cycle. We'll be talking about things like mitosis, the cell cycle, and mitosis. Okay, so before we even get into any of the study guide questions, let's examine something that I think we very quickly covered in class, which I think is very important. Okay, what are cells? According to Google, quote, the smallest structural and functional unit of an organism, cells, are typically microscopic and consist of a cytoplasm and a nucleus enclosed in a membrane. That's a lot of words. In short, cells are the building blocks of life. If you've taken a chemistry class before, or you're currently in chemistry class, and your chemistry teachers told you atoms are the building blocks of life, well, they're wrong, because you're a biologist. So, think like one. Cells are units, okay? They're quantized units. They're kind of defined things. And what are they defined by? They're defined by a cytoplasm, a nucleus, and a membrane. What's a membrane? A membrane, if you might remember from the first midterm, is a phospholipid bilayer, which is a really fancy way of saying it's a wall of cells that separate 2 environments. It's like your house. The walls of your house is like a phospholipid bilayer. It's like a membrane. It keeps stuff out and it keeps stuff in, right? When you turn on the heat in your house, you don't want all that heat leaking outside. And of course, you don't want that cold air leaking inside. So that's what a membrane's for. The nucleus is the most important part of this chapter, and it's probably one of the more important parts in this midterm, so we'll be focusing on that a lot. The nucleus is an organelle. It's a structure which contains all the genetic material of the cell. Okay? It holds all that stuff in, all those genes and chromosomes and things like that. The cytoplasm is everything else. If you've ever heard of things like the mitochondria or the endoplasmic reticulum or all these other weird things, those also are part of a cell, but we're not going to be looking at them this chapter or the next chapter or the chapter after next. So now that we've kind of got a base idea of what a cell is, we want to look at the genetic material of a cell, right? This is all the stuff that's inside of the nucleus. So it's inside the genetic material. You'd expect genes, and you'd be right. Okay? So from a macro perspective, from a big perspective to a small perspective, we have the genome, which is the biggest, the chromosomes, which is a smaller set of stuff, the DNA, which is even smaller, and the genes, which are even smallest. Okay, the genome is a set of all the genetic information contained within an organism. Okay, I'll give you a more specific definition in a short bit. Chromosomes are smaller packets of genetic information. Humans, for example, have between 23 to 46 chromosomes. Apparently, lettuce has 18. And some other animals have more. Some animals have less. DNA, deoxyribonucleic acid, is the stuff that makes up chromosomes. It's the stuff that you hear talked about in the news all the time. It's made up of genes and nucleotides and other stuff like that. Fascinating stuff, really. Genetics is so cool. We're not going to talk about it in this chapter, though. So, what is a genome? Okay, a genome is a set of all the chromosomes in a haploid cell. Now, what is a haploid cell? We'll talk about that in a little bit. But look at this picture here. If you're having trouble identifying a chromosome, you should have paid a little bit more attention in class. But here is a chromosome. See this number 1? That's number 1, and that's a chromosome right there. That's also another one. A genome is all of one of these chromosomes. So we have 23, including the X, chromosomes in a female human. And a genome is all the ones on the left-hand side, for example, like that. Okay? This will make a little bit more sense as we keep on going on, so please bear with me. A chromosome, as you can see beautifully on the picture on the right, basically looks like a really messy hair tie, or those hair scrunchies. On the left-hand side, if you look at that beautiful artist's rendition, okay, it's basically kind of this H-shaped thing that contains a whole bunch of DNA. Remember when I was talking about chromosomes being packets of genetic information? The DNA holds all your genes together, or it's a compilation of all your genes, and all that DNA gets squiggled up into the chromosomes. So your chromosomes kind of hold the DNA together. They kind of package the DNA in a nice, really complicated and complex and really fascinating way, which we're not going to talk about either. But, you know what chromosomes kind of look like. When we talk about chromosomes and genomes and all this stuff, we'll want to be talking about gametes versus somatic cells. Okay, this is what I was talking about between haploid and diploid. And we're going to refer to this information again in future slides, but I want to give you guys a quick overview right now. Gametes are reproductive cells. These are normally the cells that go out and turn into baby. Okay, like sperm from a male or an egg from a female. Those are both gametes. The unique point of gametes is that they only contain one set of chromosomes per cell, right? They only contain the left-hand set of chromosomes, for example. There's only 23 in a human, okay? And that's what we call a haploid. You can think of haploid like half, because maybe regulars too, okay? So one set of chromosomes per cell. Somatic cells, or diploid cells, is everything else in the body. And they contain 2 sets of chromosomes per cell. Or they can contain up to 2. And that's 46 in humans. Okay? I know it's a little bit kind of broad, general. Keep on trucking along and follow me and we'll get through this. Let's talk about the cell cycle. Yes, I'm skipping the second question because the second question is kind of out of place. So what is the cell cycle? This is a really good image of the cell cycle, actually, and I took this from Wikipedia. All the sources for my images are kind of inconspicuously located on the bottom, as you can probably tell. And this is a really fascinating slide. We're going to be sticking on this slide for a little bit. Okay, and this is a good representation of the cell cycle. These guys over here, these are cells. I know they don't look like, they kind of look like purple googly eyes. Just make believe that these are cells, okay? And pretend that the cell cycle starts here, kind of at the 12 o'clock position where you'd expect a clock to start. If you look on the outside circle, okay, that's what we're going to look at first. This entire orange color with the letter I stands for interphase. Okay, interphase consists of about 90% of a cell's life, sometimes even more. Okay, and then you get this like this orange interphase, and then all of a sudden you hit this yellow M. What does this M stand for? Ooh. This M, ladies and gentlemen, stands for mitosis, and it's the only reason why we're talking about this chapter. Mitosis is super cool, super interesting, and we're going to be talking about that a lot in future. Just know for now that mitosis is this division of cells. It's kind of how cells reproduce and multiply. It's really important. And during interphase, the cell is just doing its own thing. It's growing, it's helping other cells around it, it's maybe making proteins, it's doing its own thing. It's having a grand old time. It's just growing up. And mitosis is when it becomes an adult and has babies. Okay? Let's take a look at the inner circle. Okay, so we start from 12 o'clock again, and we progress along this light blue. And we're still in interphase, by the way, you see. But this part of interphase is what we call G1, or growth phase 1. Okay? And that's when the cell starts growing up. It gets a little bit bigger, gets a little bit bigger, gets a little bit bigger. Note that there's this kind of G0 phase as well. We'll talk about this in a very, very later slide. So ignore this for now. Just remember that G1 happens. There's some stuff that's growing. The cell's growing. It's happy. And then all of a sudden, the cell hits a checkpoint. It thinks, hey, I need to start thinking about making more cells. So it's going to enter into a process called synthesis. And in synthesis, this is also a super interesting part. And that's where things, that's where what we call DNA replication happens. It's when DNA replicates itself, and it's probably one of the most amazing things you'll ever see in biology. And unfortunately, we're not going to be talking about that either. But take it on my word that stuff happens during synthesis. A copy of the DNA inside of the cell is produced. And then we hit G2, or the growth phase 2. Okay? And that's when the cell keeps on growing and it's getting ready to enter mitosis. What's mitosis again? That's a good question. Mitosis is cell division. Let's look to Google for an answer. Mitosis is, quote, a type of cell division that results in two daughter cells, each having the same number and kind of chromosomes as the parent nucleus. In short, mitosis equals to cells cloning themselves. In shorter, mitosis equals to cell division, perfect cell division, reproductive cell division. Okay? Mitosis is a process in which one cell becomes two cells. And when it becomes two cells, both cells should be, in theory, identical. Let's take a look at how mitosis works, shall we? Remember, we had the different phases, and the phase right before mitosis was growth phase 2. And remember, growth phase 2 was part of interphase. In growth phase 2, we already have double the amount of chromosomes. We have 46 chromosomes in the cell. 46. Okay? And they're all kind of mixed up. I don't know if you can see this very clearly, but it's kind of just a whole big blob of stuff. In fact, we don't even have chromosomes yet, right? As you can tell, there's not even any H-shaped things. There's no kind of wiggly long things. All that is just a whole tangle of stuff, and this whole tangle of stuff turns out to be DNA. And also notice during the G2 phase that there's kind of this envelope, or this kind of membrane, and this is what we call the nuclear envelope. And this is what the nucleus is. This entire thing is a nucleus. It contains all the genetic information of a cell. So, that's growth phase 2. What happens next? Well, if we follow the arrow over here, we can see that there's a couple of cool things happening. Okay, look inside the nucleus. All those kind of spaghetti strands of DNA that's messy all over the place, they've kind of become this kind of nice, long, H-shaped thing. And what are those H-shaped things again? What do we call them? Oh, yeah, we call them chromosomes. And in fact, we call each kind of half of the H a chromatid. And because they're exactly alike, we call them twin chromatids. No, I'm kidding. We call them sister chromatids. I don't know why we didn't call them twin chromatids. We call them sister chromatids. Sisters don't have to look alike. But no, that these chromatids are should be exactly the same as the other. Okay? And there's two of them. Also note that there is this kind of thing right here where it's a little bit darker. And in fact, if you remember the other, the hair scrunchy chromosome picture, they kind of met in that middle part, right? And that middle part's actually that dark part. And that we call that dark part the centromere. Okay? The centromere is the part of the chromosome that links the sister chromatids together. And it plays a really important part in future phases. Okay? This phase, by the way, is called prophase. I don't have a witty thing for you to remember what prophase is. Just remember that the first phase is prophase. You can also see now that you get these lines that are coming out from these strange T-like star shapes. Okay, this star shape, I know it doesn't look like a star. In a video I'm going to show you in a little bit, they kind of do look a lot more like stars. These stars are called asters. Okay, and an aster is the structure that they kind of act as like anchors for these wires to hook onto. And we'll look at what these wires do in a little bit. The next phase is prometaphase. And prometaphase is actually kind of easy to remember because it's between prophase and metaphase. We'll look at metaphase in the next slide. So obviously it makes a lot of sense just to smush both pro and meta together to make prometaphase. And prometaphase is where a lot of cool stuff begins to happen. It's where you can really see mitosis happening. Because in prometaphase, the nuclear envelope or the nucleus actually kind of starts dissolving. It really does break apart into a whole bunch of different fragments. Okay, and then you get these like lines that come out from the asters, and these lines are actually microtubules. Okay, these microtubules hook up to the centromeres. What were the centromeres again? Ah, yes, the centromeres were the part of the chromosome that links both of the sister chromatids together. Okay, the microtubules, or these wires, hook up to the chromatids or the centromere via the kinetochore. See what I mean about a lot of memorization? You just have to memorize this stuff. Okay, kinetochores are kind of these structures which act as kind of a bridge between the microtubules and the centromere. In fact, they're basically the same thing. So after all those microtubules hook up to those centromeres, okay, then we're going to get something called metaphase. And metaphase is really amazing. It's when all of these, chromosomes, we have 23 pairs of these chromosomes in a human, they all line up along this line. Okay, we call this line the metaphase plate. Okay, and they line up, they really do line up in a pretty straight line. And at this point, these asters have kind of moved to opposite poles of the cell. And you can see all the microtubules, they stuck to the centromeres. So once everyone's lined up, that's the end of metaphase, and then the ending happens. Anaphase happens. Remember what the goal of mitosis is. Mitosis is the division, the perfect replication and division of a cell from one cell into two cells. So we need to make two cells that are duplicates of each other. So how do we do that? Well, we pull them apart. All the microtubules start retracting, okay, from either side, and they start pulling back to the poles, these ends. And now these chromosomes have grown up from sisters. They've become daughters. I don't see how this makes sense. You just gotta memorize this, okay? The chromatids become daughters, and they become actual full-on chromosomes. They become their own grown-up, happy little cell. And they pull away from each other. And when they keep on pulling, they keep on pulling, they keep on pulling, they keep on pulling, and eventually what happens is they pull so far apart that there's nothing left in between. In fact, there's a lot of empty space. And as these chromosomes are being pulled back towards each respective side of the cell, a cleavage furrow or a division is happening, which basically splits the cell straight in half where the metaphase plate was, and it cuts the cell in two, and it makes two new cells. And then the nucleus comes back. The nuclear envelope starts forming again very slowly, and the chromosomes get to relax. And if you remember, at this point, Then the cell enters G1, or growth phase 1, and then the cells just kind of float off and start growing on their own. That's how mitosis works. I'm going to go back one slide, and we're going to go through this really quickly just to reinforce the point. Okay, mitosis, you start with having 46 chromosomes. Okay, you get the 46 chromosomes because the DNA replicated itself during synthesis phase during the cell cycle. Okay, then you get prophase. During prophase, the mitotic spindle starts forming. The asters, or the star-shaped guys over here, they start moving into position. The DNA turns into, magically, these chromosomes, which are composed of 2 sister chromatids, which are perfectly identical. Okay, then you get prometaphase, where the nucleus dissolves, or the nuclear envelope dissolves, sorry. Then the microtubules start kind of stretching out and trying to latch on to the kinetochore, or the centromeres. Okay, and then after that you get metaphase, and metaphase is when all the chromosomes line up on a line like ducks. The microtubules are attached, and the microtubules start pulling, and that's anaphase. And those chromosomes get pulled apart, and the chromatids get separated from one another. They're so sad to see their sister go, and they become daughters for some reason, and they become daughter chromosomes. And as they're getting pulled away, they get pulled away, they get pulled away, and then you get telophase and cytokinesis, when the cleavage furrow forms and the nucleolus, or the nuclear envelope, starts coming back, and the microtubules get cut, and the chromosomes are trapped in their jail of the nuclear envelope. And then the next step is growth phase 1, where the cell, now cut in 2, begins growing bigger and bigger and bigger until it was its original size. Let's watch this in action. Okay, this is a little bit fast, but it's a really cool video. The cell that we're looking at, if it's not obvious already, is this thing in the middle. Okay, and you can see other cells on the side here that really aren't undergoing mitosis. You can see this one beginning as the middle one ends. Okay, you can see in the non-mitosis cells, like this one, that there is a very clearly defined area, right? This giant red stuff, and it turns out this red stuff is DNA. What they've done is I'm guessing here, they've put some type of dye that dyes the DNA, and under a fluorescent microscope they can actually see the DNA glow red, which is really cool stuff. Okay, so these are really red, and you can see it's all the DNA is just floating around randomly within. You can kind of see the nucleolus right here, and just a ton of DNA just chilling. But these guys aren't undergoing mitosis. This middle guy definitely is. So let's watch him. Let's go back to the beginning and see all that stuff, all that cell membrane, that nucleolus, that nuclear envelope just dissolves, right? There was definitely a defined barrier, and then just disappears. Okay, what phase is that? Do you remember? That's the prometaphase. Okay, if you also take a look, all the cells, all the DNA, it's actually easier to see in this cell right here. All the DNA, which is kind of like this mush, this spaghetti mush of colors, starts forming into these clearly defined chromosomes. What's that called? Prophase. You see how that red stuff starts forming into these defined bits, and there's more black stuff? That black stuff is the absence of DNA. So we have prophase. We have prometaphase, when the nuclear envelope starts going away. And let's look at metaphase. Can you identify where the asters are, or where the mitotic spindles are, or where the microtubules are? Remember, the microtubules are these kind of lines that pull, that kind of attach to the centromeres and pull the chromosomes away from one another. And you can see here these bright green lines that just hook onto the chromosomes and yanks them apart. And you can see how, I mean, really amazingly, how during metaphase the chromosomes are just all perfectly lined up. I mean, they line up and they literally just stay there for a little bit, perfectly lined up. It's really cool stuff. Well, what do we call it when the microtubules pull away? Right? We call that anaphase. When the chromosomes are being pulled away from one another, that's called anaphase. And they literally are. You can see that for every one of the chromosomes on the left, on the bottom side, one of those goes to the top. And actually, you can see right there, just wait for like 2 seconds, you'll see it again, the cleavage furrow happens right where the metaphase plate was. Okay, you see how there's a line right there, and then boom, that cell cuts in half. And what's that called? It's called telophase in cytokinesis. And then you can see the the nuclear envelope reforming as a DNA from the chromosomes just kind of relax. Okay? And they become kind of half the size of their original size, but they become two cells that are perfect copies of each other. This stuff is super cool. I could watch this all day. So, that's in animals. In plants, you basically have the same thing, except instead of a cleavage furrow, okay, you know, you can tell why it's called cleavage furrow. Cleavage furrow, if you have problems remembering. Instead of a cleavage furrow, plants have basically what we call cell walls. And cell walls, plant cells don't really split in half very easily. They're made out of this very resilient stuff called cellulose. It's like the stuff that makes paper. That stuff doesn't biologically break apart very easily. So instead of having to like rip the cell in half, what they do is they actually grow a new cell wall between the two new nuclei. Okay? I don't expect that you'll have to know too much other than this. Remember, this is a broad overview. So, why do we care about mitosis? What's so important? Okay. Well, mitosis is really important because of this awesome logic. Okay, without mitosis, there's no growth. Remember, there's no growth because the cells aren't dividing. You're not multiplying. If there's no growth in the organism, then the organism ceases to exist. It dies. If there's no organism, then that organism doesn't survive. According to the idea of survival of the fittest, organisms without mitosis, then they ain't fit. Organisms who ain't fit don't have children, according to Darwin, and thus, if you have no children, your genes, your genetic line, your species will not be carried on anymore. It'll be lost forever. So if you didn't have mitosis, you'd be really depressed. I'm not really sure why this question's on here. Just know that mitosis is important to evolution, or it's significant, because it's basically what drives life. It's how life grows. It's how life exists, continues to exist, and continues to grow. It's why a small baby turns into a medium-sized baby, which turns into a very loud child, which turns into a small adult, which turns into a real adult, which turns into an old person who turns into dead. Okay? That process is driven by mitosis. So we're coming back to the question that we skipped on the study guide. We're going to look at some light micrographs of dividing cells and tissue and explain what's happening. Okay, well, do we remember the different phases of mitosis? What's the first phase again? Prophase. What happens during prophase? The DNA kind of bundles up and it turns into chromosomes. Well, let's find some let's find some cells that have tons of DNA in them. That's a cell with tons of DNA. 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 Let's find a cell that still has a nuclear envelope, right? It still has that circle of stuff, but has distinct chromosomes forming. Well, let's look here. This looks like a very nice, lovely target. This looks like prophase. It's a nice, big, fat, juicy cell. And you can see the difference, right? This cell has these weird squiggly lines. It has these lines, almost black lines. And it has a lot of white space. Whereas the cell next to it has a whole bunch of small little dots and very little white space. This is a good sign that prophase is happening. What's the next phase? Prometaphase. What happens during prometaphase? Well, the membrane, the nuclear envelope around the genetic data, starts to break apart. The microtubules, or the little wires, come down and try to start attaching to the chromosomes by way of the kinetochores, I believe. Yeah, the kinetochores. And where do we see that? We can see that here is really messy. You can see that there's no defined barrier anymore, right? Here, it's kind of barrier. There's kind of gray stuff, right? There's a definite circle. Here, not really a circle. There's less gray stuff. All that gray stuff going away means that the dye, or the stuff inside, has kind of floated off into the rest of the cell. Okay, and you can see some of these lines, and we call these microtubules. What's the next step? Metaphase. What's important about metaphase? Well, metaphase is when all the chromosomes line up in a line. Do you see any chromosomes that do that? I see a few. There's one right here. There's one right here. There's one right here. Unfortunately, this picture isn't so great. We can't really see the microtubules very well, but you can definitely see the chromosomes lining up. What happens after a metaphase? Well, anaphase happens after metaphase. What happens during anaphase? Well, those chromosomes are being pulled away from each other. Those sister chromatids are being pulled away from each other. They're crying, and they become daughter chromosomes. And we're going to see that happening here, 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 here. Whole bunch of places. Okay? It's pretty distinctive. Now, the next step, which I don't know why we don't have to know, but we should know, is telophase and cytokinesis, is when the cell forms again. And you can kind of see here, these are two smaller cells, right? You can see that they still kind of have chromosomes in here, and they're just kind of dissolving into DNA. And this cell wall is very faint compared to other cell walls like this one or this one, and the cells are much smaller. I would hazard a guess that these two cells are newly formed. Cool. So we basically covered everything that we need to cover according to the study guide. I also want to cover something about cancer, because cancer is a really good example of mitosis gone wrong. This is our good buddy, the cell cycle again. Cancer is caused, or some cancer, cancer is really complicated, but there are some types of cancer which are caused by problems in mitosis. Okay, and G0, growth phase 0, is basically like the cell will enter this loop between growth phase 0 and 1, where it doesn't really do anything. Remember, synthesis is where you get replication and stuff, and DNA stuff. DNA magic happens, right? Between growth phase 1 and growth phase 0, nothing happens. And sometimes if the body doesn't need more cells, it's going to just lock the new cells in this kind of rotating phase. And if it locks it into this rotating phase, then you're not going to get new cells because it's not going to go to mitosis. Well, sometimes some cells will be a little bit altered. They'll be a little bit weird. We call them cancerous. And the cell just doesn't care. It just goes basically from mitosis, and it like blitzes through interphase and is like, nope, 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 nope. Make stuff, nope, 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 nope. And then mitosis, and then make stuff, and then mitosis, and then make stuff, and then mitosis. And it keeps on going and going and going and going and going and going and going. And you get tons and tons of cells uncontrollably. If you want to learn more about cancer, by all means, there's a lot of resources out there. But cancer is a really good example of mitosis gone wrong. So that's, mitosis is really important, but it can also hurt you. That kind of sums it up for chapter 12, I believe. This video is a little bit longer than I planned. Hey, what can I do? Hopefully this helps you guys. If you have questions, post your questions on the forum. A lot of people can answer them. Good luck studying, and stay tuned for the next part, which I believe is meiosis.